very welcome back to my channel Mis.Medicine Dr. Garima, and today we will discuss about cell structure and function, and this is very basic topic and I want to record this video 3 to 4 years back. So आज मौका मिला है, let's discuss this. So first of all, what is cell structure? In 1655, the English scientist Robert Hooke coined the term cellulae for the small box-like structures he saw while examining a thin slice of cork under a microscope. Next है basic cell structure. All cells have the following basic structure: plasma membrane, cytoplasm, and genetic material. A thin, flexible plasma membrane surrounds the entire cell. The interior is filled with the semi-fluid material called the cytoplasm. Also inside are specialized structures called the organelles and the cell's genetic material. Next आ जाता है generalized eukaryotic cell. It includes plasma membrane, cytoplasm, organelles, nuclear envelope, and nucleus. Next is the animal cell. This is also very important diagram for neat preparation. It contains polyribosome, actin filament, trophy-R, centriole, mitochondrion, lysosome, microtubules, plasma membrane, and every one have the different function. So membrane function. All cells are surrounded by a plasma membrane. Cell membranes are composed of a lipid bilayer with the globular proteins embedded in the bilayer. On the external surface, carbohydrate groups join the lipids to form glycolipids and with proteins to form glycoproteins. These functions as cells' identity markers. Next is fluid mosaic model. This is also a very important question for the NEET exam. In 1972, S. Singer and G. Nicholson proposed the fluid mosaic model of membrane structure. It contains extracellular fluid, intracellular fluid, transmembrane proteins, cytoplasm, cholesterol, carbohydrate, glycolipid, glycoproteins. Next आता है phospholipids. It contains glycerol, two fatty acids, and phosphate group. Phospholipid bilayer. Mainly two layers of phospholipids. The non-polar tails point inwards and the polar heads are on the surface. Contains cholesterol in animal cells. Is fluid, allowing proteins to move around within the bilayer. This is the structure. Mid part is the non-polar hydrophobic tails and the side ones are polar hydrophilic heads. Next is the steroid cholesterol. It affects on membrane fluidity within the animal cell membrane. Membrane proteins. A membrane is a collagen of different protein embedded in the fluid matrix of the lipid bilayer. Peripheral proteins are appendages loosely bound to the surface of the membrane. Integral proteins penetrates the hydrophobic core of the lipid bilayer, are often transmembrane proteins completely spanning the membrane. And the question is, what is the function of the cell membrane? It regulates the passage of substance into and out of the cells and between cells, organelles, and cytosol. It detects chemical messengers arriving at the surface. It helps in linking adjacent cells together by membrane junctions and anchor cells to extracellular matrix. Next is the what are the six major functions of the membrane proteins. First one is the transport, second one is enzymatic activity, third one is the signal transduction, cell recognition, fifth one is the intercellular joining, and the sixth one is the attachment to the cytoskeleton and extracellular matrix. Next आता है what is the function of the plasma membrane proteins. Transporter, it helps as enzyme, cell surface receptor, and cell surface identity marker, cell adhesion and attachment to the cytoskeleton. Next, membrane transport. The plasma membrane is the boundary that separates the living cell from its non-living surroundings. In order to survive, a cell must exchange materials with its surroundings, a process controlled by the plasma membrane. Materials must enter and leave the cell through the plasma membrane. Membrane structure results in selective permeability. It allows some substances to cross it more easily than others. The plasma membrane exhibits selective permeability. It allows some substance to cross it more easily than the others. Next is the passive transport. Passive transport is diffusion of a substance across a membrane with no energy investments. There are four types of passive transport: simple diffusion, dialysis, osmosis, and facilitated diffusion. Solutions and transport. Solution is homogeneous mixture of two or more components. It can include solvent and solutes. Solvent means dissolving medium and solute means component in smaller quantities within a solution. Next is the intracellular fluid, which include nucleoplasm and cytosol. And extracellular fluid includes interstitial fluid and plasma. Interstitial fluid, fluid on the exterior of the cells within the tissues. Plasma, fluid component of the blood. Diffusion, the net movement of a substance from an area of higher concentration to an area of lower concentration down a concentration gradient, which is caused by the constant random motion of all atoms and the molecules. Movement of individual atoms and molecules is random, but each substance moves down its own concentration gradient. Here is the lump of sugar example. Lump of the sugar is added in the water. Random movements leads to net movement down a concentration gradient. Then all sugar is dissolved in the water. No net movement at equilibrium is present. Diffusion across a membrane. The membrane has pores large enough to a molecule to pass through. Random movements of the molecules will cause some to pass through the pores. This will happen more often on the side with more molecules. The dye diffuses from where it is more concentrated to where it is less concentrated. This leads to dynamic equilibrium. The solute molecules continue to cross the membrane but at equal rates in both directions. Diffusion across a membrane. Then how diffusion occur across a membrane? Two different solutes are separated by a membrane that is permeable to both. Each solute diffuses down its own concentration gradient. There will be a net diffusion of a purple molecules towards the left, even though the total solute concentration was initially greater on the left side. Next, the permeability of the lipid bilayer. Permeability factors: lipid solubility, size, charge, presence of channels and transporters. And there is hydrophobic molecules which are lipid soluble and can pass through the membrane rapidly. Polar molecules do not cross the membrane rapidly, and the transport proteins allow passage of hydrophilic substances across the membrane. How passive transport processes. There are three typical special diffusion that involve movement of materials across a semi-permeable membrane. It includes dialysis, which is selective diffusion of solutes, facilitated facilitated diffusion, and osmosis. Dialysis, selective diffusion of solutes, have lipid soluble materials and small molecules that can pass through the membrane pores unassisted. And in facilitated diffusion, substance require a protein carrier for passive transport. And osmosis is simple diffusion of water. Now, what is osmosis? Diffusion of the solvent across a semi-permeable membrane in living systems. The solvent is always water, so biologists generally define osmosis as the diffusion of water across a semi-permeable membrane. Osmosis is a lower concentration of solute, higher concentration of sugar. Then there is same concentration of the sugar in the second image. And there is selective permeable membrane. Sugar molecules cannot pass through pores, but water molecules can. More free water molecules means high concentration. And the other part means right hand side, water molecules cluster around sugar molecules. Fewer free water molecules and there is lower concentration. Osmosis, water moves from an area of higher concentration to the lower concentration. Osmotic pressure of a solution is the pressure needed to keep it in equilibrium with pure H2O. The higher concentration of solutes in a solution, the higher its osmotic pressure. Tonicity is the ability of a solution to cause a cell to gain or lose water based on the concentration of the solutes. If two solutions have equal solutes, they are called isotonic. If one has a higher solute and others and lower solvent is hypertonic, and the one with the lower solute and a higher solvent is hypotonic. Water balance in cells with the walls. This is the plant cell, which is turgid firm and generally healthiest in the hypertonic environment, where the uptake of water is eventually balanced by the elastic wall pushing back on the cell. Definition of osmosis is the diffusion of water across a semi-permeable membrane from a hypertonic solution to a hypertonic solution. This is very easy. Next is the facilitated diffusion, means it need a special protein to transfer. Diffusion of solutes through a semi-permeable membrane with the help of a special transport proteins, that is large polar molecules and ions that cannot pass through the phospholipid bilayer. That's why there are two type of transport proteins can help ions and large polar molecules diffusion through cell membranes. First one channel proteins and the second one is the carrier proteins. Channel proteins provide a narrow channel for the substance to pass through, and the carrier proteins physically bind to the substance on one side of the membrane and release it on the other side. मीन्स पकड़ के रोड क्रॉस कराते हैं एक तरह से. इट मीन्स दे आर हेल्पिंग टू क्रॉसिंग रोड्स. They are specific, passive, and saturates. In specific, each channel or carrier transports certain ions or molecules only, and in passive, there is direction of net movement is always down the concentration gradient. And the third one is the saturates. When all transport proteins are in use, rate of diffusion cannot be increased further. Active transport, which means they need energy to move from one place to another. It uses energy, means ATP form. Energy is equal to ATP to move a substance against its natural tendency up a concentration gradient. Requires the use of carrier proteins, transport proteins that physically bind to the substance being transported. There are two types, membrane pump and another one is the sodium potassium pump. Membrane pump, protein mediated active transport. A carrier protein uses energy from ATP to move a substance across a membrane up its concentration gradient. Next one is the sodium potassium pump, one type of active transport system. So how does it work? First step is the cytoplasmic sodium binds to a sodium potassium pump. Then sodium binds stimulates phosphorylation by ATP. Phosphorylation causes the protein to change its conformation spelling, expelling sodium to the outside. And then extracellular potassium binds to the protein, triggering release of the phosphate group. And then loss of phosphate restores the protein's original conformation, and potassium is released and sodium sites are receptive again. The cells repeats. Next is the coupled transport. There are two stages, carrier protein and coupled transport protein. Carrier protein uses ATP to move a substance across the membrane against its concentration gradient, which is storing energy. Next one is a coupled transport protein, which allows the substance to move down its concentration gradient using the stored energy to move a second substance up its concentration gradient. Here is the review of the passive and active transport compared. Next is the bulk transport, which allows small particles or groups of molecules to enter or leave a cell without actually passing through the membrane. There are two mechanisms of bulk transport, is endocytosis and exocytosis. What is endocytosis? The plasma membrane envelops small particles or fluid, then seals on itself to form a vesicle or vacuole which enters the cells by phagocytosis, pinocytosis, or receptor mediated endocytosis. There are three types of endocytosis we already discussed. Cytosis means in phagocytosis, a cell engulfs a particle by wrapping pseudopodia around it and packaging it within a membrane enclosed sac large enough to be classified as a vacuole. The particle is digested after the vacuole fuses with the lysosome containing hydrolytic enzymes. Next is pinocytosis. In pinocytosis, the cell gulps droplets of extracellular fluid into tiny vesicles. It is not the fluid itself that is needed by the cell, because any of all included solutes are taken into the cells. Pinocytosis is non-specific in the substance it transports. And what is the process of phagocytosis? First step, chemotaxis or adherence to microbe to phagocyte. Second, ingestion of the microbe by phagocyte. Third, formation of the phagosome. Fourth, fusion of the phagosome with the lysosome to form a phagolysosome. Fifth, digestion of ingested microbe by enzymes. Sixth, formation of residual body containing indigestible material. 7. The discharge of the waste material. And here is the receptor mediated endocytosis. Next is exocytosis, the reverse of endocytosis. During the process, the membrane of a vesicle fuses with the plasma membrane and its contents are released outside the cells. There are cell junctions also. Tight junctions, desmosomes, and gap junctions. Cell junctions means long lasting or permanent connections between the adjacent cells. There are three types: tight junction, desmosomes, and gap junctions. Tight junctions, the membrane of the neighboring cells are very tightly pressed against each other, bounded together by the specific protein, forming continuous seals around the cells. Tight junction prevent leakage of extracellular fluid across a layer of the epithelial cells. Next, what are the desmosomes? Desmosomes, also called anchoring junctions, function like rivets, fastening cells together into strong sheets. Intermediate filaments made of sturdy keratin proteins anchor desmosomes in the cytoplasm. Third one is the gap junctions, also called communicating junctions, provide cytoplasmic channels from one cell to an adjacent cell. Gap junctions consist of special membrane proteins that surround a pore through which ions, sugars, amino acids, and other small molecules may pass. Gap junctions are necessary for communication between cells in many types of tissues, including heart muscle and animal embryos. The next part is the nucleus and the nuclear envelope, repository for the genetic material called chromatin, which contains DNA and proteins. Nucleolus holds chromatin and ribosomal subunits, region of intensive ribosomal RNA synthesis, and nuclear envelope, surface of nucleus bound by two phospholipid bilayer membranes and double membrane with pores. And the next one is the nucleoplasm. Semifluid medium inside the nucleus is nucleoplasm. Next is the chromosomes. DNA of eukaryotes is divided into linear chromosomes, exist as strands of chromatin except during cell division, histones, associated packaging proteins. Ribosomes are RNA protein complexes composed of two subunits that join and attach to messenger RNA, site of protein synthesis assembled of nucleoli. Endomembrane system. It compartmentalizes cell channeling passage of molecules through cell's interior. Endoplasmic reticulum, they have rough and smooth. Rough endoplasmic reticulum studded with the ribosomes and smooth endoplasmic reticulum have few ribosomes, which make proteins. Rough endoplasmic reticulum is especially abundant in cells that secrete proteins. As a polypeptide is synthesized on a ribosome attached to rough it is threaded into the cisternal space through a pore found by a protein complex in the membrane. As it enters the cisternal space, the protein folds into its native conformation, and rough is also a membrane factory. Membrane bound proteins are synthesized directly into the membranes. Smooth endoplasmic reticulum is rich in enzymes and play a role in a variety of metabolic processes. Enzymes of smooth endoplasmic reticulum synthesize lipids, including oils, phospholipids, and steroids. These include the sex hormones of vertebrates and adrenal steroids. In the smooth of the liver, enzymes help detoxify poisons and drugs such as alcohol and barbiturates. And smooth endoplasmic reticulum stores calcium ion. Muscle cells have a specialized smooth endoplasmic reticulum that pumps calcium ions from the cytosol and stores them in its external space. When a nerve impulse stimulates a muscle cell, calcium ions rush from the into the cytosol, triggering contraction. The Golgi apparatus is the shipping and the receiving center for the cell products. Many transport vesicles from the endoplasmic reticulum travel to the Golgi apparatus for modification of their contents. The Golgi is a center of manufacturing, warehousing, sorting, and shipping. The Golgi apparatus consists of flattening membrane sacs, cisternae, and looks like a stack of pita bread. Next one is the Golgi sorts and packages materials into transport vesicles. Function of the Golgi apparatus we already discussed. Next is the membrane bound organelles. Lysosomes, vacuoles, and peroxisomes. These are very important organelles and very important topic for the exams. Lysosomes vesicle containing digestive enzymes that break down food and foreign particles, vacuoles, food storage and water regulation. Peroxisomes contains enzymes that catalyze the removal of the electrons and associated hydrogen atoms. Mitochondria, sites of cellular respiration, ATP synthesis bound by double membrane surrounding fluid-filled matrix. The inner membrane of mitochondria are cystae. This is the powerhouse of the cell. The matrix contains enzymes that breaks down carbohydrates and the cystae house protein complexes that produce ATP. Cytoskeleton. The eukaryotic cytoskeleton is a network of filaments and tubules that extends from the nucleus to the plasma membrane that support cell shape and anchor organelles. Protein fibers, actin filaments helps in cell movement, intermediate filaments, microtubules and centrioles. Centrioles are short slender with the 9 plus 0 pattern of microtubule triplets. Centrioles may be involved in a microtubule formation and assembly during cell division and in the organization of the cilia and the flagella. Cilia and flagella helps in the movement, contains specialized arrangements of microtubules are locomotory appendages of some cells. Cilia and flagella share a common ultrastructure. Cilia, small and numerous, and flagella, large and single, have 9 plus 2 pattern of microtubules and are involved in cell movement. Cilia and flagella move when the microtubule doublets slide past one another. Each cilium and flagellum has a basal body at its base. Cilia to and from motion. I hope you like this video. Do subscribe the channel. Bye bye. Take care.